There we go. So Martha, whenever you're ready.
Yep. Thank you so much, Michaela. Good morning, everybody, and thank you so much for joining us in one more of our WWN webinars. Today, we have the pleasure of having Dr. Rachael Dangarembizi, which is a senior lecturer at the Department of Human Biology and a neuroscientist in the Neuroscience Institute at the University of Cape Town in South Africa. She has established the first laboratory in Africa that studies the mechanisms of brain injury caused by fungal neuroinfections. Her research bridges basic science with clinical insights, aiming to improve diagnosis, treatment, and patient outcomes by understanding fungal host interactions, neuroimmune responses, and brain injury. Dr. Dangarembizi leads several organizations dedicated to advancing neuroscience capacity across Africa with the aim of growing local expertise and infrastructure to drive impactful research. Today, she will present to us the webinar Exploring Updates in Neuroinflammation. Of course, this is an important topic since inflammation has actually been implicated in several neurological diseases. Rachael, welcome and thank you for being here.
Thank you so much, Martha. Thank you. Would you like me to share my screen or did you want to?
Yes, go ahead.
Announce anything? Okay. All right, so I'll go ahead and do that. Just let me know when you can see it.
Yes.
Yep.
All right, fantastic. So good afternoon, morning, depending on where you are, evening. It's afternoon where I am. I'm in Cape Town in South Africa, and my name is Rachael Dangarembizi. I am a neuroscientist who's interested in fungal infections of the brain. I will not be talking about fungal infections today. Maybe fortunately or unfortunately, I'm not sure, but I will be talking about neuroinflammation in health and disease. So I would like to start off by congratulating WWN on your 15th anniversary. I just learned today that WWN is 10-15, and it's 15 years of standing on the right side of scientific history. And we are super proud of you, WWN. Thank you for supporting women in neuroscience to advance their careers. I'm one of those that have benefited from your work. And so on behalf of other women in neuroscience, thank you so much, and well done to the leadership, well done to the community of women, as well as to the sponsors for making this possible. And then one last sort of little announcement as well I'd like to sneak in is the fact that the IBRA World Congress will be in Africa for the first time in 2027. So it will be coming to Cape Town. And I would like to invite all of you, if you can, all roads lead to Cape Town in 2027. Be sure not to miss this. All right, now we can get into our task for the day. So I was thinking when I was preparing this lecture, I was like, what would be the best approach to take? Because neuroinflammation is such a broad topic. And one could go in several ways, looking at the different diseases that are applicable to neuroinflammatory symptoms, or different ways that neuroinflammation can affect the brain, etc. But I thought that one of the most sort of misunderstood concepts is actually how neuroinflammation comes about, both at the organ level, at the cellular level, at the molecular level. And most people find it a little bit intimidating when they look at all of those signaling charts with hundreds of molecules and arrows going in every direction. So I thought that maybe what we could do in this 40 or so minutes is to discuss this at a very basic and very conceptual level. And then if we understand that, we could apply it to any kind of disease or any kind of neurological disorder that we work on. So I've put here a little outline of how this lecture will go. I'll start off with an introduction of the CNS immunity, just talking about the CNS immune landscape in general. That is the big picture. How does the CNS deal with immune insults? And then after that, we would then go to molecular mechanisms underlying neuroinflammation. How does it actually occur? What is it? And what are the molecules at play? And then after that, I will go on to key mediators of neuroinflammation. That is looking at the different cell types or the major players that mediate the process of neuroinflammation. And lastly, we would have then a consideration of physiological versus pathological effects of neuroinflammation as a sort of a close to the discussion that we'll be having. So to start us off, on the path of learning about CNS immunity, I have decided to take us a little bit back to the history that shapes what we know about CNS immunity today. And this started very well long back in the 17th, 18th century when neuroscientists were becoming interested in knowing how the brain deals with insults, deals with infection, etc. And one of the key landmark experiments that I just wanted to point out is this experiment by Edwin Goldman that eventually, led to the discovery of the blood-brain barrier. I'm sure all of us know about the blood-brain barrier, but it was backed off of work developed by a guy called Paul Ehrlich, who injected trypan blue into the nervous system and discovered that it actually didn't go anywhere. It stayed quite compartmentalized within the CNS, that is within the brain, the spinal cord, and with extensions to the retina of the eye. And this led to the belief that the CNS as an environment is actually quite set apart or isolated from the rest of the body, which is true, and which led to other further developments that I'll talk to just now. In the middle of the 20th century, around 1948, you have more experiments from Medawa and colleagues who were doing experiments to see how the immune system of the brain responds to tissue that is grafted. So they took tissue, they would graft it into the periphery, and they would get immediate rejection, because in the peripheral immune system, anything that is deemed foreign is immediately rejected. But when they put that in the brain, they discovered that the brain was quite quiet for a long time, actually, months. This tissue could stay in there and there would be no inflammatory response. And so that led to the development of a concept that was known as immune privilege, sort of the belief that the brain is in a state of relative immune tolerance, that it tolerates insults in a way and is incapable of raising an immune response. And so that concept of immune privilege, unfortunately, prevailed for quite a long time and was quite misleading until quite recently. We've discovered quite a little bit more about the CNS immunity that I'll talk to now. But basically, immune privilege had people thinking the brain was something like that, like a fortified castle that is surrounded by water and has very little connection to the outside world, which is the periphery in this case. And there was so much evidence to support that. Part of it being the fact that there was no clear lymphatic system, like what you see in the peripheral immune system. There was a limited number of immune competent cells. You couldn't see any T cells and B cells in the brain, at least then they couldn't. And the cells in the brain also lacked these MHC molecules for antigen presentation, which made it seem like indeed it's an immunosuppressed environment. And then lastly, this knowledge that there's a blood-brain barrier sealed that immune privilege concept, and people started thinking there's actually no communication with the peripheral immune system. So, as I said, now we know differently. And what we know from the turn of the 21st century is that, number one, the brain is actually not a homogeneous environment. It is compartmentalized. It is made up of different compartments that behave very differently from an immunological perspective. You have the parenchyma, which is basically the area where you find neurons and glia, and that is relatively immunosuppressed, and for a reason. I'm sure all of you will agree with me. We don't want too much inflammation in the brain because we've got neurons, and neurons are amitotic. They don't divide by mitosis. That means that the ones that we're born with are the ones that we're going to die with, basically. And so to protect them at all costs, we try to dampen down the immune response, at least in the parenchyma. But that doesn't mean that there's no immune responses anywhere else. We've got other compartments like the ventricular system, where we make CSF, and it is very, very responsive to immune insults immediately. We also have the meningeal compartment, which is made up of membranes that surround the brain, and it is also very immunologically responsive. So we have learned quite a lot in terms of how the CNS as an environment responds to outside insults. And there've also been few discoveries recently that show that we actually do have a histologically distinct meningeal lymphatic system. We also have immune competent cells within the brain, and then the CNS is capable of mounting an inflammatory response and producing inflammatory mediators. And lastly, that there is communication between the CNS and the peripheral immune system. So these 4 points are basically what shapes our lecture today. How the brain responds to insults in an inflammatory fashion. So as I said now, we know that there is active immune responses in different compartments of the brain, and that the brain is, is capable of mounting an inflammatory response. And what we also know is that the brain has got these barrier systems that are helpful, number one, for ensuring that there is immune surveillance in the CNS, but at the same time that immune cells from the periphery do not get into the parenchyma and cause hyperinflammation for reasons we've already mentioned. So if we're looking at this image, try and get a pointer here, looking at this image, we've got the meningeal layers that are between the skull and the parenchyma. And in there, you've got dural vessels that are actually fenestrated, or they've got pores around them, which means that they allow cells from the periphery to come out and to do surveillance of pathogens, et cetera, et cetera. But then that surveillance ends right there, because if you go further down, we've got the pia mater, which is the innermost layer of the meninges. It has got tight junctions that ensure that those cells will not traverse into the parenchyma. That's the first barrier. And then the second barrier we have is at the level of blood vessels that sort of supply the cerebrum. These vessels penetrate into the neural tissue, but they are surrounded by what's called the blood-brain barrier. I'm sure all of us know about it. It's made up of astrocytic end feet as well as basement membranes. But what the blood-brain barrier basically does is to ensure that no cells that are circulating in the blood vessels will be able to cross over into the parenchyma unless if needed. And then also no pathogens that could be coming in from the circulation will cross over into the parenchyma. In that way, we are protected. And then the last barrier we've got is at the level of the CSF forming organs, which is known as the choroid plexus. And there we have tight junctions again that ensure that no pathogens, no blood cells will cross over into the parenchyma. So at the end of the day, what we have is the brain quite protected from immunological insults by cells that remain quite outside of the parenchyma. They are surveilling, but they cannot get into the parenchyma unless if it is really, really necessary. So the question then arises, who takes care of the parenchyma? Now, there are two cell types that are in the parenchyma that are known as immune resident cells of the brain, microglial cells and astrocytes. And these two cells are important for dealing with any pathogens that breach all of these barrier systems that I've talked about and end up in the parenchyma. Now, if a pathogen gets into the parenchyma, astrocytes and microglial cells will try to deal with it. And if they cannot, they also have the option of recruiting cells from the periphery and getting them to be shunted into the parenchyma to deal with it. So overall, and in summary, what I'm saying is that the CNS as an environment is not necessarily immunologically silent. It is actually very active. Number one, it has got cells that are surveilling around and looking for pathogens, but not necessarily in the parenchyma. And then it has got barrier systems that ensure that pathogens are kept at bay and do not get into the parenchyma. And lastly, it has got resident cells within the parenchyma that ensure that if anything crosses in, it can be dealt with. So that's basically the summary of what we know now about CNS immunity. Now that brings us to the discussion of the day, which is about neuroinflammation. Now that we know that the brain is capable of mounting an inflammatory response or an immune response, we now want to define what neuroinflammation is. Neuroinflammation is defined as an inflammatory response within the brain and the spinal cord that is mediated by cytokines, chemokines, reactive oxygen species, or secondary messengers of different types. All of these are just mediators or communication molecules released by cells within the brain and the spinal cord. Within the brain, as I said, we've got astrocytes, microglia, and when they're activated, they release these cytokines or chemokines, and that basically is what we call neuroinflammation. It is quite confusing for quite a number of people to understand this concept of neuroinflammation, because at the back of their mind is usually the description of inflammation in the general immune system. Peripheral inflammation or general inflammation usually involves circulating leukocytes being brought to sites of infection, right? Let's say you've got an infection in the lung, you're going to get leukocytes being brought to the lung, and then they will, within the lung, deal with that infection. In the brain, there's no need necessarily in the first instance to have recruitment of leukocytes. You can actually have neuroinflammation without recruitment of cells from the periphery. Why? Because you've got resident immune cells like astrocytes and microglia. And then these resident cells release inflammatory mediators, as I said, cytokines, chemokines, and the basic function of these mediators is to induce, number one, gene induction or gene transcription for release of, for triggering biological functions. I will talk to that in a little bit, but basically, inflammatory mediators like cytokines will trigger biological responses in their target cells, and we'll look at that. But they will also cause proliferation and migration of other immune cells. So for example, if an astrocyte is activated by a pathogen, it will release cytokines so that other astrocytes can start multiplying and coming to the site of infection, or other microglia can multiply and come to the site of infection. And so that is also a biological effect. And then lastly, the concept of neuroinflammation also confuses biologists a little bit, because they always think inflammation is maladaptive or negative and associated with neurodegeneration. But neuroinflammation is actually a physiological process that occurs even if there's no necessarily damage. So it can happen just under normal physiological processes. It helps to deal with homeostatic disruption, and after that, everything goes back to normal. So it's not every case that we end up with damage or neurodegeneration. That's the point we're making. All right, so that is the definition of neuroinflammation. And then we go on to the causes of neuroinflammation. Neuroinflammation is caused by a number of things. In fact, anything that causes disruption in CNS homeostasis will result in neuroinflammation. It doesn't necessarily always have to be a pathogen or something from outside. It can be internal disruptions of homeostasis within the CNS. Changes in ATP levels, changes in solute levels, buildup of waste, cells that are dying within the CNS, all of those things will trigger a neuroinflammatory response. And here I've also listed other external causes that may also cause neuroinflammation. That includes toxins from the environment or environmental factors. You've got obesity and metabolic disorders causing neuroinflammation. You've got genetic variations or polymorphisms, mutations, etc., causing an inflammatory response in the brain. You also have autoimmune diseases that create autoantibodies that may trigger neuroinflammation. There's infections, oxidative stress as a result of infection or other mechanisms. There's dysregulation of cells within the brain, such as astrocytes and microglia. All of that may lead to neuroinflammation. And recent evidence has also shown that you can also have maternal immune activation, either as a result of infection or alcohol consumption or any other things, being sort of a cause for neuroinflammation in the developing fetus. And that may actually lead to neurodevelopmental disorders. And then lastly, you've got all sorts of drugs and xenobiotics that could be circulating. If they cross over into the brain, they're able to cause neuroinflammation. In summary, neuroinflammation is caused by anything that can disrupt the homeostatic environment within the brain. So what we've done so far in the last 7 or 8 minutes is to talk about the general CNS immune landscape. What cells are there, what triggers inflammation, and what inflammation is. But now we get to the slightly intimidating bit, which is molecular mechanisms underlying neuroinflammation. And as I said, this usually intimidates people because there are so many signaling steps that go on in the process of neuroinflammation. But to help us navigate this, I've decided to dumb it down to the lowest level possible for us to understand it and then build it up from there. In summary, before we get into everything, in summary, neuroinflammation is just basically based upon the recognition of what's called danger signals by a specialized type of receptor on an immunocompetent cell, which then triggers internal cellular processes that lead to a biological response by that immune cell. Simple 3 steps: the stimulus, recognition, internal signaling, and then a biological response. So all of the 101 steps that you then see in signaling molecules are basically just, details of those 3 basic steps: recognition, internal signaling, and a biological response. And if we understand that, then it will be very easy for us to sort of understand what happens within the brain when a pathogen arrives. So now I've brought in a little bit of detail into that last figure that we had. And here we have a blood vessel that is supplying the brain, and we've got the parenchyma where we find neurons, glial cells, etc., etc. On the left-hand side, in the blue region, under normal circumstances, when homeostasis is maintained, all of these cells are resting, they are happy, neurons are happy, and they're carrying out their usual functions of generating electrical signals. And that is kept well and good because we've got this blood-brain barrier that has got tight junctions and nothing can get in from the blood vessel. But then if you bring in a pathogen, like a fungus, in this case, a notorious fungus that is able to break down the blood-brain barrier, what you end up with is a leaky blood-brain barrier that allows pathogens to get in. And suddenly we now have these pathogens in the parenchyma where they are actually not needed. And then neuroinflammation kicks in. What happens is that our 2 populations of resident immune cells will, through the expression of receptors on their surface, detect the presence of this foreign invader, and they will quickly release cytokines. And then the cytokines will then lead to a biological response. It can be phagocytosis, antigen presentation, complement activation, all of which are meant to just help with clearance or killing of that pathogen. So that's basically the major steps there. These resident immune cells, if they are overwhelmed, They can also release what are known as chemokines or chemoattractants, which are basically chemical molecules that help to call for help from peripheral cells that are circulating in blood. And then these will now come in and they will also help produce cytokines and clear the fungus. So in summary, neuroinflammation occurs when there's a danger in the form of a pathogen or also in the form of anything internal that is disruptive, and that is detected by resident immune cells. And then that is then dealt with. After all of that is done, under normal circumstances, all of these cells will reset and all of these cells will either be taken back into the circulation or else they will they will kill themselves by apoptosis. And so that is the process of inflammation in the presence of a pathogen. Then comes the question of cytokines and what they do. What are cytokines? We've already said they're communication molecules that are released by these resident immune cells. But their effect is basically on other cells, as I said. They can cause proliferation, but they can also activate other cells to induce a biological effect. So let's say a microglial cell had released a cytokine. That cytokine will bind on another cell, which is the target cell in this case, cause gene activation in that cell, and then we'll have a biological effect. That target cell has got to have a receptor for that cytokine, basically. Now, a lot of us do research and misuse and abuse cytokines, unfortunately. Why? Because cytokines are a little bit confusing. So I've brought this question of what do cytokine levels actually tell us? What do the concentrations of cytokines in the brain tell us? Does it always indicate the fact that there is inflammation and that it is bad and it has to be treated? And the answer is no. There was this very quirky statement that I saw that says cytokines are like letters of the alphabet. They are meaningless if they are not used in context. So just seeing a cytokine raised like IL-6 does not necessarily mean that, there's neuroinflammation suddenly, number one. Two, it is bad, we've got to reduce that level of IL-6. No, that's not the case. Cytokines should be taken within the context within which they are coming, because they're actually very complex. Two things make cytokine function a little bit complex to understand. The first thing is called pleiotropy. And this is the situation where the same cytokine has got diverse, but sometimes unrelated biological effects. Mm-hmm. So seeing a cytokine in the CNS does not necessarily mean that you're going to see the effect that you think, that cytokine causes. It could cause a completely different effect depending on timing. And I'll give an example. When there's an inflammatory insult, like a pathogen, TNF is one of the early cytokines to be released. Within 2 hours, you've got an increase in levels of TNF. And that early signal is aimed at propagating the inflammatory response to the pathogen. And then after some time, TNF levels will go down. And then later on, maybe after about 16 hours, you may get TNF levels coming up again. This time for reducing the inflammatory response or regulating their inflammatory response. So now if you were doing an experiment and you measure TNF at 2 hours, you can say, we've seen raised levels of TNF, therefore there's neuroinflammation and we need to treat for it. If you measure at 16 hours, raised levels of TNF, therefore there's inflammation. But in these two cases, TNF actually has very different biological effects, and it is all about timing. The second complexity is what's called redundancy, where different cytokines can mediate exactly the same or similar effects. So seeing a number of cytokines raised does not mean anything unless if we take it within the context of how they are expressed. I've put here a puzzle word set or something like that to just say what we do sometimes in biology is to just go and say, I'm looking for the cytokine R if we were taking cytokines as letters. I'm looking for the cytokine R. As long as we find it, then there's inflammation. But that presence of that cytokine could be meaningless because it's not making, it doesn't have any context to it. In some cases it does. It makes a word that you can read. In some cases it doesn't. It's just a cytokine that is raised and has got no context to it. All right. I hope I didn't confuse you with that. But it's not every case, is all I'm saying, that we see cytokines, that it means we've got neuroinflammation and that it is bad. The effect of cytokines on their target cells is also a little bit of a mini complex thing, but I've tried to summarize it in this little diagram here, where the cytokine producing cell releases a cytokine and it binds to receptors on its target cell. It also triggers internal signaling, which eventually leads to gene transcription within the target cell. That gene transcription can lead to all sorts of biological effects. It can be phagocytosis, or it can trigger release of other enzymes or cytokines, et cetera, that is dependent on the target cell. But what we can do is that we can use some of these steps as readouts for seeing if there is actually inflammation within the brain. And we've done that quite successfully, at least in our lab. So one of the things you see in the target cell that has been targeted by a cytokine is that there is upregulation of the receptors to that particular cytokine. And that can be a sign that that cell is activated. Number two, we can also look at signaling molecules and how they are, they are activated within that target cell. Some of them actually move from certain areas of the cell to others, like this example that I've given here, which is known as NfL6. It's usually in the cytosol or in the cytoplasm, and it's a transcription factor. When it is not activated, when there is no cytokine or anything that has activated the cell, NfL6 is sitting in the cytosol. But once that cell is activated, then it will translocate and go into the nucleus. So that event of translocation from one cell part to the other can be taken as a signal or as a readout for activation of that cell. And then lastly, you can also have transcription of genes, which means that you can use sequencing, for example, to see if a cell has changed its gene transcription patterns. And lastly, the release of other molecules like cytokines as well. So to summarize this second part of this lecture, we've talked about the molecular mechanisms underlying neuroinflammation. First thing, 3 steps only. There is recognition of a danger signal by receptors on the surface of a cell, and then that triggers internal signaling, and there's a biological effect. And that is exactly the same thing that happens with cytokines. They bind to a receptor, there's internal signaling, and then that triggers a biological response. Now, we move to key mediators of neuroinflammation. And I know that I've sort of intentionally flipped these around. Rather than starting by telling you about astrocytes and microglia and things like that, I thought to sort of just go at the basic level and say, these are the mechanisms at the molecular level. Now we can go to the cell. The CNS is, as we said, furnished with 2 resident or main resident immune cells, which are microglial cells and astrocytes. So we'll start with microglia. Microglia are developed, or rather originate from the periphery, actually. They come through the circulatory system and migrate into the CNS during embryonic development. For humans, that's around 4 weeks post-gestation, 4 weeks into pregnancy. And then for mice, that's probably around embryonic day 7 to day 9 or something. So quite early during embryonic development, you have migration of microglial cells, which are hematopoietic. They are made in the embryonic yolk sac, and they migrate to the CNS. After that, migration, they start differentiating and maturing into what we call mature microglia. And this happens before birth, actually. By the time a child is born, they already have their full complement of mature microglia ready to fight infection. And these mature microglia have the capacity to renew themselves. They are self-renewing, and they therefore keep the population of microglia going for the rest of our lives, basically. Experiments that have been done using what's called fate mapping show that there's no new recruitment anyway during the course of life. The microglial cells that we'll have had during embryonic development are the ones that we keep, and they keep themselves replicating themselves to renew the population. The same population that we have will also start aging with time, and that sometimes causes issues and disease because they become hyperactivated or primed, and sometimes they become old and their response is kind of compromised. In terms of function, these microglial cells, they are physiologically important because they mediate learning and memory functions by releasing neurotrophic factors, for example. They are involved in pruning synapses. They are involved in clearing up cellular debris and unwanted proteins and solutes. They are involved in, phagocytosing, invading pathogens, etc. But at the same time, besides the homeostatic or physiologic effect, they may also have pathologic effects, especially if they're aging or if they're primed. Sometimes they get hyperactivated, they start destroying synapses, or they can start generating unwanted proteins. Sometimes they start actually destroying viable neuronal cells. And a misconception that has been carried for quite a long time is this aspect of dichotomous polarization of microglial cells. And I know this is a very contentious issue, and I'm happy for us to discuss it a little bit in the discussion coming up. But people have believed that microglia exist in two forms, the M1 form and the M2 form, M1 being the classically activated, they're hyperinflammatory, they release pro-inflammatory cytokines, and M2, they are more physiological, they release anti-inflammatory cytokines. This has been shown by recent research to be not entirely correct. And another thing as well is that M1 is usually seen to be amoeboid in structure, therefore secular. So it tempts people when they do histology and they're looking for neuroinflammatory indicators. When they see microglial cells that are nicely rounded, then they say they were all activated, therefore we had neuroinflammation. That is not necessarily the case, because if you look at the inflammatory pattern or morphology rather of activated microglia, you will see that it is a continuum. It is a spectrum of activation. It starts off in the ramified sort of side, going all the way to the amoeboid, but there's quite a lot of different morphologies in between that can be either quiescent or they can be activated. So if you see a microglial cell with these processes around it, it doesn't necessarily mean that it is not activated. It could actually be activated, but just in the spectrum of activation. And I bring that, a little bit further to ways of measuring microglial activation and basing on morphology and whether that helps or not. There are quite a lot of studies that have been done that sort of say, yes, you can measure what's called fractal analysis, which is basically branching patterns of your microglial cells, and that will tell you whether they're activated or not. Or else you can look at what's called skeletal analysis, which looks at the branch length, how long do they branch out of the cell body. You can look at cell body perimeters, like in this case, how big is the cell body? Is it rounded? And how much is the area? And then lastly, you can do what's called the Scholl analysis to look at the complexity of branching, and that will tell you whether, it is ramified or whether it is amoeboid. This paper by Green et al. Is a really cool paper for anybody that wants to use morphology to go and look at the weaknesses of each of those analysis patterns. It is not necessarily always the case, and they don't always tell the same story. They may be able to show the sort of morphological shift at the population level of how microglia have shifted, but they do not necessarily indicate that microglia are activated. I would like to share within the last probably 5 minutes of our lecture how we, in our lab, for example, look at microglial activation. I'd like to pose this as a potential for other labs as well that want to look at microglial activation and to measure it. If you remember from our molecular mechanisms in neuroinflammation, we talked about the fact that once there's recognition, there's going to be signaling that goes on within the cell that eventually leads to release of cytokines. Now, if we took advantage of that signaling, for example, the movement of transcription factors from the cytosol into the nucleus, and we used those events as functional readouts, we would actually be able to tell apart activated cells versus non-activated cells. Now, I have given this example. This is a hippocampal slice with so many cells inside it that are activated using LPS. And what we did was basically to stain for the nuclear translocation of one of the transcription factors that we know is triggered in interleukin-6 signaling. That transcription factor, which is known as NFL6, once there is a signal, either a pathogen or a cytokine, it moves from the cytosol and goes into the nucleus. So any cell that you see expressing that transcription factor in the nucleus is activated. And I would like you to look at how different an activated slice looks from an inactivated slice. If you've got a control and there's no inflammatory signal, there's actually literally no translocation of that transcription factor. So in the end, if you use a pipeline for counting the number of nuclei that have got that transcription factor versus those that are not, you can actually come up with the numbers of cells that are activated versus those that are not. And that helps rather than just showing, there's red, therefore there's activation. You can actually quantify the number of distinct cells that are activated or not. So using that, you can also co-stain that nucleus with an inflammatory, or rather a cell type marker like IBA1 for microglia, for example. And in this case, we show that these were actually microglial cells that were activated because they co-localize the nucleus that is activated with the cell type specific marker. And to buttress the point that I made earlier on, that when you look at microglial cells and their morphology, it's tempting to think that the moment you see them rounded, it means that they're activated. In our work with fungal pathogens, we see microglial cells that are very well amoeboid in structure, but not activated at all, which is paradoxical, but it makes the point I was trying to make. So what we have here is normal microglial cells with their ramifications stained by IBA1. And then here we've got microglial cells that have been infected by this fungus that is known as Cryptococcus, which is appearing in red. And as you can see, they've phagocytosed all of that fungus and it is sitting inside them. But if you try to measure whether they are activated or not by measuring that NFL6 translocation that I was talking about, you will see that actually they do not have any translocation of NFL6 into the nucleus to show that they're not pro-inflammatory in their nature. And if you try to measure the types of cytokines that they release, you will also see that there's basically no change in inflammatory cytokines in these microglial cells. You can also go further as to even look at gene expression. Here we use single nuclear RNA sequencing to look at genetic changes in these microglial cells in response to the fungus. And what you see is that in the crypto-treated, there's absolutely nothing. But if you treat with LPS, there's upregulation of inflammatory genes. This is work done by some of the team members in my lab who are probably also in this room. But that's that about microglia. And I will speak one last minute about astrocytes as the second cell type. Astrocytes discovered very long back, the time that we described neurons in the CNS already by Golgi, around the turn of the 19th century, astrocytes were already sort of spoken to. And you find them in very early drawings of Golgi. By 1883, he had already figured out that they form the components of the neurovascular unit. But astrocytes, as we know, they've got pathogen recognition receptors, and they are able to respond to danger signals by releasing chemokines or cytokines. And we've already spoken to the molecular mechanisms of that. In terms of morphology, if you want to measure activation or neuroinflammation in astrocytes, they actually distinctly change their morphology. They become bigger, they also multiply in numbers, and they increase the expression of a protein that is known as GFAP. So if you are to measure GFAP intensity, you can actually see that it increases in astrocytes that are activated versus those that are not. You can also look at pathogen recognition receptors like Toll-like receptor 4. They're activated. You can also look at internal signaling molecules like NfL6. Lastly, you can also look as well at gene expression. And this is also work from our lab where we show that there is a change of genetic transcription in astrocytes that are infected with cryptococcus compared to control, and that it is slightly comparable to what you get when you stimulate with LPS. So those are the two main resident immune cells, but perhaps to end this off, I would like to talk about the fact that there are other unappreciated immunocompetent cells within the CNS that also contribute to the neuroinflammatory response. And one of those is endothelial cells, or cells that make up blood vessels. They're really important because they're the first contact basically with pathogens that are circulating. And if you stain for inflammatory markers, you can actually see that they also get activated. So they serve to tell the astrocytes and microglia that there are danger signals coming your way. And this is work that I have done. You can look up some of our papers that show that there's increased immunoreactivity and gene expression in endothelial cells. I think I'm left with very little time, but probably end off by saying it's very tempting to want to separate all of these cell types in a dish and have microglial pure cultures and then do your experiments on that. But typically in the brain, they would actually talk to each other. So what you end up with when you separate these cells and start using them in isolation is that you lose the crosstalk between them. And the response that you see is not necessarily representative of the actual response that would happen within the brain. And that has been shown elegantly by this study here, which I'm not going to go into, that just looked at comparisons of microglia that were grown in a dish or in vitro versus microglia that had been freshly isolated from a mouse brain. Mm-hmm. What you see is very different, distinct gene expression patterns. So think about it when you set up your experiments and consider the fact that there is usually crosstalk. These cells talk to each other and influence each other, and they work in synergy. I think I will end here, and then the rest of the concepts can come in our discussion. I'd like to leave some time for that. Thank you.
Thank you so much, Rachel. This was very, very educational because there is so much misinformation and misinterpretation of data when looking at inflammatory, molecules like cytokines. I know a lot of people study things in the brain and they say everything causes inflammation because I actually, tested BDNF and it's high. And yeah. I think that we need to really stop doing that and start thinking about understanding what it is that we're actually doing to the brain, understanding what are the cells that may actually be affected by what we're doing to the brain, whether we're adding a toxin, whether we're adding a medication, whether we're actually doing a structural thing. And it's really imperative that we think we stop doing experiments and start thinking about, what is that we're really looking at and what it is that we really think we should be getting out of, of how we're manipulating the brain. So thank you so much. This was really, really great.